DNA. The basic monomer of DNA are nucleotides. Nucleotides are made up of one phosphate group, sugar, and nitrogen base. As going along, nitrogen bases adenine, thymine, cytosine, and guanine attach together using hydrogen bonds so that they are easy to split in DNA replication. The bonds between the nucleotides are covalent bonds so that the outside structures stay and do not break. DNA is synthesized by using different types of enzymes. These enzymes include DNA polymerase, DNA helicase, ligase, and primase. Here, DNA helicase goes and splits through the DNA and cuts the hydrogen bonds. Next, behind the ligase is DNA polymerase, placing down new nucleotides that are to be added to that are going to be the new strand to DNA. DNA polymerase can only go from 3 to 5. Once one new strand is completed, polymerase must now turn back around and go 3 to 5 the other way. Polymerase is still placing new nucleotides on the other strand to make a complete strand of DNA. Once finished, there are 2 new sets of DNA, each containing one piece from the original DNA.